There we go. Well, welcome everyone. We are super excited today to have another great member of the Choroideremia Research Foundation team, Dr. Michael Young, sponsored by Genentech. With their help and their, as I say, their support, we are getting funding and assistance and encouragement to continue these great educational webinars. And Dr. Young has been with the CRF for quite a long time, and he's been to several of our conferences, but couldn't make it to the last one that we held. So he is awfully nice to join us today. We spoke last week and you were in the middle of writing up or finishing some grants. How did that go?
Well, mixed bag. I got most of them in, but one of them turned out to be an ineligible faculty member, so we had to pull the plug on one of them, but that's pretty good because another one popped up today, a big one. So we're looking to repopulate the laboratory as we've kind of gone the road of a startup company to kind of get our therapies into the clinic. I need to fill my lab up again, so it happens once in a while.
We sure hope that happens quickly. Well, listen, before we get started, I just want to again welcome everybody. We do have a chat feature, folks, that that's part of my job, is just to do the electronic part and then to monitor the chat. And as questions come in, maybe as you stop periodically, Michael, I'll just ask you the questions that are coming in. We'll have some time at the end, as most everybody always likes to ask additional questions. But today, everybody, we're going to be learning more about Dr. Michael Young's work on rods and cones and the evolution of stem cells in this process, discussing hopefully the concept of using stem cells with finding a CHM treatment cure eventually, hopefully. But this is going to be great information for us to take home. Again, we'll be recording it. And I think we should go ahead and let everybody know who doesn't know you come from Mass Eye and Ear. You have an appointment there. We've seen you several times as you were lecturing there in Boston. And you also are the director and co-director of several different important, I can read them, I want to make sure I get them right. Co-director of the Ocular Regenerative Medicine Institute and director of the Gunzburg Center for Retinal Regeneration.
Very good.
Hey, there you go. Well, I had it written down, so I got it right. So thank you for joining us. And again, everybody, the chat feature is working, use that. And again, thank you so much for joining us and taking time out of your schedule on a Monday afternoon. We really appreciate it.
My pleasure.
Mm-hmm.
Okay, now the important part. Can I share my screen?
It's working.
Okay, now, but I can't see what's going on. Okay, so I need to go here.
There you go.
And here, can you see one slide now?
Sure can. Thank you.
All right, now I'm in my own way. All right, welcome everybody. So yes, I'm going to talk today about work in my lab for the last about 20 years or so involving stem cells for the treatment of inherited retinal diseases. I'm going to do my best to at all times bring up the relevance to CHM. And briefly what that involves is, from a stem cell perspective, is, in my opinion, the transplantation of two different cell types. Because in CHM and choroideremia and diseases like it, both the retinal pigment epithelium, a support cell in the back of the eye, and the photoreceptors, the rods and the cones, are affected. And so we need to attack this problem stepwise, such that we can eventually, we hope, transplant both of these stem cells or the progeny of these stem cells, so that we might be able to restore vision in patients who have lost vision due to these diseases. Most of my work now in, let's say, the early stages targets retinitis pigmentosa, where we're targeting either the rods or the cone cells, or both. But many people around the world, as you may know, are using retinal pigment epithelial cells to treat macular degeneration. And so this is the early stages of this work. In the future, we'll be using tissue engineering techniques to transplant both of these cells. And this will be applicable to both macular degeneration of the cone variety and CHM. And I'm not advancing. There we go. Okay. So conflicts of interest are important to note. If we're going to commercialize this and bring it to the market so that we can treat patients, we have to have intellectual property. So I have patents that are owned by Mass General Brigham involving most of the things I'm going to talk to you about today. And there's a startup company of which I'm a founder. I'll tell you a little bit, called InGel Therapeutics, where we're trying to bring light to patients who have lost vision due to the loss of cones. So as many of you know, there are a lot of people in the world suffering from retinal disease. It's estimated that 250 million people worldwide have a retinal degenerative disease. And that includes macular degeneration, CHM, and other types of retinitis pigmentosa. So broadly speaking, RP, as it's known, typically involves a mutation in your rods. So rods are the cells that die first. About 1 in 44,000 patients in the US. And it typically comes on early in life, from 10 to 30 years. And there's no effective treatment for most forms, although there is a treatment for retinal pigment epithelial 65 protein known as Leber's congenital amaurosis. That's gone very well. Less so well for other diseases so far. And we can talk a little bit more about the gene therapy trials in CHM if you wish. So ways to treat these diseases. Of course, maybe 10 years ago, there were no treatments for any of these types of retinal degenerative diseases. And that's changed now. So there is certainly, as you know, gene therapy, neuroprotection with growth factors. There was a chip that was implanted into the eye for types of retinal disease. That seems to have gone away now. Optogenetics is a new field. Maybe some of you have heard about it. That's where one takes a gene therapy approach to make a cell that is not photoreceptive respond to light. This is really cool stuff. So when all your rods and cones go away, scientists have figured out that they can take the protein from bacteria or other critters that live in the sea and make, for instance, your ganglion cells or your bipolar cells, either other retinal neurons, make them respond to light. And we can talk more about that if you like as well. What I do is cell replacement or cell rescue with stem cells. And we have a pretty simple approach. For most of the work you're going to hear about today, the principle is pretty consistent. We use retinal progenitor cells. So these are cells from the retina that can become lots of different cell types. They're not really stem cells, but they're progenitor cells. So they can't make kidney or pancreas, but they can make eye cells. And that's to our benefit, if you can imagine. Typically, we use fetal donor eyes in the early stages. And we do that for a couple of reasons. One is that embryonic stem cells and induced pluripotent stem cells you may have heard of have some great potential, but they also have some drawbacks in this field. And one is that they are pluripotent at times. And that means they can make pancreas and liver. And one doesn't want that in the eye. So we think by starting with fetal donor eyes, we have a cell that's biased towards making retina and is not going to form a tumor, simply put. And so we think there's a great safety component to this that allows them to be used in early stage phase 1 clinical trials that makes them safe. So typically the problem with this approach is you can't grow enough of them. And we solved that many years ago with some great technology where we grow our cells in low oxygen conditions, not 20%, which is what we're all breathing now, but 5 or 10% now. And the max we've ever gotten is up to passage 19 without changes in the cells. And what that means is we can get trillions and trillions of cells from 2 donor eyes. And we think that makes the fetal approach translatable to the clinic. Now, the other thing that we really always insist upon is minimal manipulation for safety. And that means we don't use gene therapy viruses. We don't change the cells in any way. We just figure out how to grow them. And that makes them also very safe. So a fairly complicated figure here. I'll just take you, if you can see my pointer here, I hope, to where we want to be, which is right here in this kind of magic spot with a post-mitotic photoreceptor precursor. So why would we want a post-mitotic cell? Because it's really not post-mitotic if you grow it at low oxygen. So if you come back to one stage before to these lineage restricted progenitor cells, they can make lots of different cell types. We want a cell that's committed to making a rod or a cone, but is still able to divide if we feed it low oxygen. So our cells are actually off of this chart. So most people would say, many people in reviews over the years have said that you can't grow a rod precursor cell or a cone precursor cell. If it's dividing, it's going to become lots of different cell types. And in the animal, in a human, that's the case. But when we put them in a dish and grow them with the right low oxygen culture conditions, we can get them to grow, and we can get them to grow like crazy. So that's really enabled our work over the years. So this is what a human fetal retina looks like. There are no outer segments or outer plexiform layer. What that means is the photoreceptors are immature and they haven't formed synapses yet. So important for us is that we can show that when we get different donations or when we get different passages, meaning different expansion levels, the cells are the same. And so that has to translate into the clinic, these cells have to be consistent. And I hope you can see, without getting too much into the data, that these gene expression analyses here that we're looking at are very consistent from one donor to the next. They're all clustered around this plus or minus 1 line. So again, we spent, and by we, I mean some brilliant scientists in my lab who have gone on to great careers of themselves. Peter Baronoff at Harvard and Bud Tucker at Iowa working with me, discovered that after many years of growing cells that look like this, they grow for a little bit and then they stop, and then they stop. Here you can see 4 different examples of cells that just stopped growing after about a month. We bought a new incubator, we turned off the oxygen, and they grow like this. So this is transformational for us. It allows us to generate cells that can be used clinically. And this is what it looks like. This is a trial that we did with Reneuron, I'll tell you a little bit about now, a small biotech company in Wales, where if you start on the left with 2 retina, you grow them in these normal size flasks, they're called T75 flasks. We start with 4 of them and about 8 million cells. We grow them and grow them and grow them until we get 120 million cells, and then we seed them into vials in the freezer. Then we take one of those vials, or in this case, 2 of those vials, and we grow them in these huge T500 flasks. 1, 2, 3, 4 more passages, and then we have 2.4 billion cells. That's known as a working cell bank, and we have 400 vials of those. And again, 1 more passage and we could have 6,000 vials if we wanted to, but we don't do that. We don't want to push it too much. You don't want to have chromosome changes by growing your cells too much. But again, 2 vials out of that 400 grown and grown and grown and grown, and pretty soon we have 2.4 billion cells again. And that gives us the potential from 2 eyes to treat 900,000 patients. That's a potential. Again, we don't push it that much, but that's the realistic possibility is we could treat 1 million patients from 2 eyes. And that means not only will this treat the world's population of RP, but we can even, if we're smart enough, figure out how to treat macular degeneration as well. So just a little bit of data on what we saw when we took these human retinal progenitor cells and put them into blind mice. We put them into the subretinal space, which is this region here under the retina. Here you see a cluster of cells. They're all green in this case. They're marked with this dye PK867. But again, this mouse is blind because it has no rhodopsin, the magic molecule that make rods see. So whenever anything is in this layer here of the outer nuclear layer, we call it the rod and cone layer, that has rhodopsin, they light up. And I think you can see a lot of cells lit up here. And that means we have made new rods in this rodless mouse. It doesn't actually have no rods, but the rods that it have are non-functional. So we think we've made this mouse see again. And we test that with a rat in this case. So the Royal College of Surgeons rat, a very distinguished name for a rat, that goes blind from an RPE cell defect. So the support cell in the back of the eye goes bad, and that kills the rods and cones. A very similar model from a structure and function point of view to choroideremia. Different mutation, but in this case, the rods and the RPE die, and that leads to the death of cone. So we can test the vision of this animal by putting it into an array in which there are rotating bars around the cage. And if the animal can see those rotating bars, it turns its head. And we can calculate that right down to this number here, which is visual acuity. So it's basically an eye chart for rats. And when we do that, if we put in saline solution, we see no changes between left and right eye. But if we transplant one eye, in this case, this eye, this is the right eye, OS is left eye, OD is right eye, we see a massive change in the ability of that rat to see. Okay, so the next thing we did with these studies, and again, this is about 10 to 15 years ago, all this work took place through funding from the Foundation Fighting Blindness and this small biotech company, Reneuron. We did a study in pigs, mini pigs, in fact, a whole bunch of them, as you can see here, 40 of them. This study was done in Brazil with a former student of mine. And we tested pig cells in pigs. Now, why would we want to study pig cells? Couple of reasons, and this is important from a funding point of view if you're looking at studies that are being proposed. Transplanting human cells into an animal tells you some things, but it misses a lot of other things too, because mostly of the immune system, but there are other components too about how a cell makes connections and integrates and forms synapses that don't get along between different species. So what we did was, we call it the analogous allograft work, in which we grew a pig cell from a green pig in this case, that would allow us to compare it to the human cell and grow the pig cells that are exactly the same as much as they can be to a human cell, and then put them into a pig. And that's the best predictor of what would happen if you put human cells in a human. It's what we call it an allograft, not a xenograft between different species. So what we did was we transplanted these cells into the pig and studied what happened. So again, this is an unfortunate slide. Somebody made a mistake in the early stages and used an antibody against green fluorescent protein and turned it red. It's confused people for years now, but red is green. Think about that. Red is green here. But the important thing is these are cells all in red under the retina, and they're, some of them at least, are part of the retina, this layer here. But really importantly, this layer here shows that these cells are making connections with the host. This is what we call a plexiform layer, where connections are made. And if you look at this on the other side here, you can see, again, cells that we transplanted under the retina migrating in and becoming part of the retina, expressing markers of retinal cells, here rhodopsin, and just filling the presynaptic layer with synaptic contacts. So this tells us a couple of things. Here, another image of the same example of the same. This layer here, if you can see all these green, tells us that we're forming synapses with the host. And so for a photoreceptor to work, it has two ends. One end captures light. That's what you're seeing here. These are primitive, but still functional outer segments. These bits capture light. So it captures lights, and then it sends that information onto other cells. And that's in the plexiform layer. And so these pig cells are doing exactly those two things. So that led to a clinical trial. It really helped enable a clinical trial at Mass Eye and Ear and several other centers around the world, Oxford and, oh dear, Portland, Oregon, at the KCI Institute. And I believe also there were some patients that were injected down in Texas, in Dallas. But this is a direct injection of cells under the retina. It was an orphan indication for RP. Eric Pierce was the original PI. Jason Commander took over. And this took, I'll just summarize a few things. It started in 2016 and it ended in 2021, unfortunately. And I'll tell you a little bit about that. There's a typo here. That should be 2. This 1 should be a 2. And so I'll take you to the next slide and show you that some of the patients got back a tremendous amount of vision. 20 letters, 15 letters, 25 letters, 23 letters. So this is 4 or 5 lines on an eye chart. But not everybody got back this level of vision. Some did not. And it didn't persist for more than a year or two, depending upon the patient. Some patients it did, but some didn't. And so if you're the company in charge here, what are you going to do? You're going to try to get more. Everybody wants more vision out of their patients and have it last longer. So what the company decided to do was put in, rather than 1 million cells, put in 2 million cells. And that led to adverse events. Too many cells under the retina, in my opinion. I wasn't in charge of this trial, of course. I invented the cells, but I have to pass it on to a company to take care of the clinical trial. And I believe that they put in too many cells. And that led to adverse events that halted the trial. The trial was not halted. The company decided not to proceed with the phase 3 because of funding issues and also because of the adverse events. And so I'll take you through what this looks like here in terms of this is one of the early patients. Sorry if anybody's you can look away if you're a little queasy about eye surgery, but this is the back of the eye of an RP patient. This is the needle going in, getting ready to make a small retinotomy, a little hole in the retina. All that black pigment you can see is indicative of RP, retinitis pigmentosa. There's a little flashlight probing to try to find the retinotomy, the little hole. And I think you'll see there goes the needle. And there go the cells, I think. You'll see a bleb form. There we go. You see that little blister that formed under the retina? Those are where the cells are going to go. And now you see what's being raised on the back of the eye. Those are all cells going underneath the retina. So you can imagine if you put in too many cells, that's not good for the patient. And so I believe that's what happened is too many cells were put in. Okay. So what about these patients that got all their vision back? What was happening? I showed you pig data that is evidence that these cells, when transplanted under the retina, can make new rods, maybe even some cones. But the work that I showed you in the rat on getting their vision back probably shows preservation of host function. So we really don't know how these cells are working. Okay? So what did I do? I decided that I needed to take a little bit of control so that this doesn't happen again. And so I started working, next stage was to isolate cones, which are what's needed for RP, what's needed in one case, certainly for CHM, and certainly for macular degeneration someday. This is the team that worked on this, especially Peter Baranoff did most of the work that led to this, the low oxygen work. But I'll take you to the next stage of the work involving cone photoreceptors. So what do we do to isolate cone photoreceptors that's different than what we just did? So we use a new technique to purify the cells. So we use a gentle sorting technique that allows for dissociation and purification of cone cells, progenitor cells, rather than other organoid-based work that you may have heard about. So this produces very pure and high viability of the cones that we isolate. It also eliminates some of the other cells that are present in the mix, such as microglia, which could induce rejection of the cells. And we do this in a manner that is GMP compatible and scalable. What that means is these cells can be used in the clinic. So in this case, we're targeting diseases where cones die specifically: cone rod dystrophy, Stargardt's disease, and other cone dystrophies. So the magic here is this machine here called a Taito from a company known as Miltenyi in Germany. And this allows us to purify cells. Again, see the difference in this diagram than the one I showed you before. Now we're starting with a multipotent retinal cell, but rather than just growing them and kind of hoping that we get lots of rods, we try to get cones by purifying them based upon their markers on their surface. So there's a couple of surface markers that we turn different colors. There are lasers in this machine that pull out the cells and give us the cells we want. In this case, either rods or cones. The first data I'm going to show you about is cone data. So again, we start with a fetal retina. We break it up into single cells, and we see that they're very alive. Only the green cells are alive. There are a few dead ones. But if we look after we sort them 3 days later, 92% of them are still alive. And then this graph here shows you that they're pure as well. So we can get cones out at about 95 to 98% pure. This actually shows 94% here, but also highly viable cells. And just like I showed you before, we can transplant these cells into animal models. Here a rat. And now again, the cells are green, not because we get them from a green person like the pig, but we get them from, we use an antibody that only stains human cells. And I hope you can see down in this image here in the lower right that the photoreceptor layer of this rat has lots of human cones in it. All of these yellow cells are human cones. So one question that arises when you transplant cells in the eye is the issue of fusion. Does the transplanted cell become a new cone, or is it just transferring its cytoplasm into another cell in the host and fooling you? This panel here shows you that that's not what's happening. These are real bona fide rods and cones, in this case, cones being transplanted. So we look in this model of retinal regeneration here. This is the RCS rat again, but we're not looking at function in this case. We're looking at cells that are growing in the back of the eye, preserving the host. And you can see here now, we call it the outer nuclear layer again, the rod and cone, looking at it from OCT, an imaging system that in blue, we save some cells. The RCS rat is prone to this sham effect, but the cells that we inject profoundly protect the rods and cones from dying. So one other way we can test the function of a rat is with an ERG. Many of you have heard about that, I'm sure, but it's kind of like an EKG for your heart, only it's in the eye. So it's an electrode that's placed over the eye and you see the waveform. In an untreated eye, you get a very small wave. When you give a medium dose of cells, that turns out to be the best, you get a much better response. Here's that optokinetic or optomotor response I told you about earlier. This is the way that you test vision in this rat. These bars are rotating, and I think you can see, looking at these p-values that tell you how real the data is, how significant it is, that even a low dose or a high dose protects these cells for 60 days from dying, and the animals can see better. So when we put these cells into the eye, now of a RD1 mouse, they make cones. These are all human cones in green, human cells in green, cones in red, and almost all of these human cones are spread out over the retina in a mosaic. This is a normal, an RD1 animal that has not been transplanted. It has no cones left by 35 days of age. This animal has lots of cones, and these have all been transplanted into the retina. And just a really pretty picture that I have hanging on my wall. As you can imagine, I think this is a beautiful image. The cones seem to not clump together, but spread out in what is known as a mosaic pattern. We've done mathematical modeling of this, and they really do spread out much like normal cells would in the retina. And this allows you to contemplate repairing vision in a more functional way. So the ERG, again, that we've tested, the cone-mediated ERG, the photopic ERG, as we call it, is improved with either 100,000 cells per eye or 150,000 cells per eye. We see a much better photopic ERG. So we can test in a dish whether these cells are functional as well by testing what's known as the mean spike amplitude or the mean firing rate when we stimulate them with light. And this really cool image. I wish I could show you the movie of these cells lighting up when you shine light on them. Here's the waveform showing that these cells are really firing when they respond to light. Okay, so that's where we are with this. So this is we've patented this cone work. There's a company known as Ocular Vision that is trying to bring this into the clinic. We are not involved in that, except that we own the intellectual property at Mass Eye and Ear. And so they are out in the present trying to raise money to start a clinical trial to treat cone dystrophy with these cones. So for me, what I want to do with these cones is someday combine them with retinal pigment epithelial cells. And so we're working on tissue engineering techniques that again would be relevant to choroideremia, where rods and cones and RPE are lost, but really that central vision that's lost when the cones and the RPE are diseased, that's what's most important for the patient. And so that's the kind of thing we're writing grants on last week and writing grants on this week together too, is tissue engineering, making complex, we call them constructs that typically involve a biodegradable polymer and 2 different stem cells. And so that's very complicated, as you can imagine, and is years away, but it's not decades away anymore. But so what are we going to do next? We decided once we finish with this cone work and let it be licensed by a company, what we're going to do next. And so I really thought about what is needed and what can be translated in the near term. And I really think what can be done sooner rather than later is preserving what's there, maybe for years and decades. And so that can really be much quicker to translate, but also much more beneficial to the patient. So I've often said in the case of gene therapy that if one can save the neurons that are there in the eye, that are already connected, that's much easier than transplanting a new one. So how do we use stem cells to preserve what's there? Well, we formed a startup known as InGel Therapeutics that we're working on now to do just that. Our mission is to bring innovative cell therapy to bring light back to our patients. This is our motto that our scientists came up with. I think it's pretty innovative as well, but that's really what we want to do is restore vision by preserving vision. And that might seem like a contradiction, but when one treats photoreceptors with the treatments that we've developed, they actually work better than they did before. So not only do we make them last longer, but we improve their function. And I'll tell you how we do that. So this is the team we've assembled. This is my laboratory. Deepti Singh and Pierre Drummond were really the key people in my laboratory. They're now the CEO and the CSO of this group. Sorry, I keep doing that. Luckily for us, we found a business guy that can help us bring this work into the clinic. Somebody who's already brought a drug to Pisa into the clinic. Patrick was a student at the Harvard Business School working on his MBA after that experience. He's actually taken time off from that work to get us going, and he's doing just that now. I work with a guy named Myron Spector at Harvard and MIT, very experienced biomaterials researcher. So you see we bring biomaterials into this. And luckily for us, we've enlisted Dean Elliott, the chief of the retina service at Harvard Ophthalmology, the kind of person who, if I've heard this before, if your mother needs surgery, Dean is the one you want working on it. He's that good. And so we have this team and some business advisors that have brought us to the point now where we are off to the races. Okay, so this image of the retina, again, just brings home the same point that it's really, in the case of choroideremia, unfortunately, it's all these cells that are damaged, rods, cones, and RPE. We're targeting first a simple disease in which only the rods are damaged. But when the rods are damaged, the cones die. And so what do? We try to save the cones. How do we do that? By delivering the factors that the rods delivered. Okay, it's a little complicated, but one patient with RPEs go blind, meaning they lose all their vision, even though their rods are the only mutated cell. Why would that be? That's because the cones need the rods. And when the rods go, the cones go next. So if we don't replace the rods, what if we just replace the molecules of the rods, and then we could preserve central vision in lots of different retinal diseases. And that's our hypothesis. So we think we can contribute to early stage, late stage, and mid stage with this approach. If it's too late, meaning all the rods and cones are gone, then cell replacement, like I just described to you, or optogenetics is needed. Early stage, when all the cells are there, gene therapy is more applicable. So this is the sweet spot for us. We call it late early stage, mid stage, or mid late stage. Broadly speaking, the biggest market. So lots of things lead to cone cell death, lots of pathways. But this is the molecule here that we're really interested in, RDCVF, rod-derived cone viability factor. This is the main molecule that our cells make. So they make lots of this. I mean, 1,000-fold more than any cells I've just described to you. And so that makes them very important. They also make a lot of other things too, that deal with metabolic stress, oxidative stress, and inflammation. So our approach at InGel is to intravitreally inject. So that means behind the lens, but in front of the retina. Everything I've told you up to about now involves a subretinal space injection, which is very invasive and can lead to adverse events. Vitreal injections are very safe. So patients with wet AMD get these every month or every 2 months, and they've shown to be very safe. So what we're injecting is a cell matrix. So this is what it looks like here. Lots of different molecules of, in this case, it's gelatin and hyaluronic acid. That's exactly what the vitreous is made of, this liquid gel-like substance that's in front of your retina. So that's impregnated, if you will, with rods that we choose to get from stem cells in this case. And they secrete lots of bioactive molecules in the form of an exosome. So like little bundles of chemicals that cells secrete. That's how cells communicate. They don't really secrete molecules all by themselves. They secrete them in these little fatty balls that are actually very little. They used to be called microvesicles or nanovesicles, but they're in the order of 20 to 50 nanometers. And so they're very small. And so they're secreted by these rod progenitor cells. They're contained in this biomatrix and then slowly released to the back of the eye over months and months and months. We can do at least 6 months delivery with the system that we have now. This is a little complicated, but it just shows the cells are making these little exosomes. Because they're fatty in their covering, they get taken up by the recipient cells, in this case, cones, and they give them survival factors. So they survive rather than die. So the work that we've done to patent this, which is what's needed to bring us something into the clinic, is that we use fetal rod cells using our microfluidic technique I told you about that gives us high purity and viability. Then we look at the secretion of these exosomes. And that's a very unique constellation of molecules, about 500 molecules that we've categorized. But interestingly enough, the most abundantly expressed is the one that's most important, this rod-derived cone survival factor, rod-derived cone viability factor, isolated first by a scientist at Pittsburgh named Jose Sahal when he was in Paris. And that molecule is then secreted and contained within this biomatrix of gelatin and hyaluronic acid. And then we have, it sounds complicated, but then it just comes down to an injection. We inject this matrix into the back of the eye. We call it IGT-001, our first product. And we think we're going to be able to preserve vision. And we think that mainly because of this mouse study. So it all comes down to mice usually. Most of the models of retinal degeneration are in mice. The most difficult to repair is the RD1 mouse. It's the first one described, hence it's RD1, retinal degenerative number 1, and it goes blind in its first 4 weeks of life. So by 3 weeks of age, almost all the rods are gone, and by 4 weeks of age, almost all the cones are gone, very rapidly. People are afraid of this mouse in this field because it's very difficult to rescue. And so we tackled it first, and we tackled it by injecting it late. This animal was injected after 3.5 weeks of age, when all the rods were already gone. So we know what we're doing is preserving them because of our cells. This is what it looks like in the central and peripheral retina. So these animals don't have a macula, an area of central vision like humans and non-human primates, but their central retina is enriched in cones, and they die first in this disease. So here we show if you leave these animals alone, in the central retina, there are no cones left, 1 or 2. If we put the cells in alone, we get more cells, more cones, but if we come in with IGT-100001, we get a 10-fold increase in the number of cones in the central and a 3-fold increase in the ones in the periphery 3 weeks after injection. And I know that doesn't sound impressive to many of you may, but to me, who's been doing this for 25 years, it's remarkable. And if we compare that to what a lot of other people are doing in this field, it's just simple. No one's ever seen this before. This level of preservation is really unprecedented. It's a lot of apples and oranges because nobody uses the same paradigm, but we did it the most difficult way. Meaning, if you look at when we intervened, we did it late. And we looked at when we evaluated them, we looked at them later too. So no one has ever looked at a RD1 mouse at 50 days of age with an intervention at this stage and seen this kind of preservation, where over half of the cells are still there. So this kind of summarizes our platform. We have a highly purified cell population. We can generate rods, cones, and even ganglion cells. And we can inject them into the front of the eye, into the back of the eye, or anywhere else in the central nervous system, we think. Okay, so this really, again, a take-home message is that retinal progenitor cells, everything I told you about today really is a retinal progenitor cell. We get them from embryonic retinal tissue. We can get them from pluripotent cells as well. That's not so easy. It takes longer. They can be expanded into many trillions of cells in a dish. And like induced pluripotent stem cell-derived retinal progenitor cells, they can make rods and cones in high numbers when differentiated. But unlike these pluripotent cells, we think they're very safe and unlikely to proliferate when grafted, and that allows us to think about translating them into the clinic. And so this is the real team that worked on this. The cell counting that you saw was generated by a very talented undergraduate that's been with me since his early days as a high school student, Michael Madison. This is Pierre Drummel, the CEO and really brilliant scientist, graduated from MIT in 4 years with a PhD. That's unusual. And this is Deepti Singh, who's been with me for many years now, thank goodness. And she has the green pipette, as we say. She can grow any cell. And I will stop there. And I think I can answer questions at that point.
My, oh goodness. Boy, I'm glad you're on our team.
Thank you.
You know, we don't have anybody asking questions yet. I think people are probably thinking what questions to ask. You know, things that I wrote down, a timeline, you know, we always ask you that. And you said again, not decades, but years, you know, so I think somebody, if it's not me, is going to ask you, all right, so what do you need from us? What can we do, if anything? What is a potential timeline? And then also you talked about stages, early, mid, and late. And when is the optimal or best time for this treatment potentially with choroideremia?
Okay, so I would say, well, by necessity, clinical trials start with a phase 1 safety trial.
Mm-hmm.
Okay, and that typically means late. The FDA would say start late so you can't do any harm. But more recently, in terms of stem cells, specifically in the eye, I would say they don't want to do anything to a patient and who has no potential benefit. Okay, so that leads to pushing back a little bit into, it depends on, you know, I talked about a lot of stuff here. The last stuff that we're doing now, neuroprotection, you can't neuroprotect the cell that's not there. So We have great imaging and functional testing now that can show whether there are functional rods and cones available. And so that's the key point for that study. We would make sure that the cells are still there to be protected. And again, the rejuvenation aspect of this work, I think, is quite important to note. So I mentioned to you, if you look at this picture, can you see my screen still?
Yep, we sure can.
Okay, I'm circling this. This is a rod.
On the far
Cones
left.
are very similar. This whole bit here are the outer segment discs that capture light. So we think when you give them the right growth factors, these grow back. And what that means is you may not be generating new rods, but the rods that are there or the cones that are there work better because they have more disc and can capture more light. So that can lead to an improvement of vision, even in a patient that doesn't get new rods or cones. So you're preserving vision, yes, but you actually can make vision better with this treatment. And so it's early stages. That hasn't been borne out in patients yet, but we think that's the case. In the Reneuron case, it's very in the first trial I talked to you about, again, some of these patients got their vision back to a remarkable degree. But here's the thing. When you transplant these cells clinically, you have no idea what the fate of the cells are. They're invisible, okay? They're not green. You can't really see them in the back of the eye unless you mark them in some way. You can't tell donor from host. So if you transplant cells into the retina, you hope they disappear. If they don't, they're probably forming a tumor, and that's a problem. But it makes it very difficult to interpret the clinical data because you don't know the fate of your cells. So whether you're repairing the cells that are there or putting in new ones is unknowable. And you can believe that the company and our think tanks at Harvard, when we have these meetings, consider this a great deal. How do you study the fate of the cells without marking them? And if you mark them, are you changing their safety profile? So we need a breakthrough in the technology so we can see these cells after they're grafted, and that hasn't come about yet. There are all kinds of cool techniques that have been developed using iron nanoparticles and things like that, where you could image them, but I don't think a phase 1 safety trial really wants iron nanoparticles involved in it. So it's a challenge, I would say. Now, as far as when, the first trial has come and gone, I would say. I don't see it being restarted. The cone work is probably 3 years away from the clinic. I would say it's very advanced. They're doing manufacturing now. The work that I'm doing now with InGel, we just had a meeting with the Foundation Fighting Blindness and had a really good discussion about when, and we're about 3 years away from our first patient, if funding goes well. Now, when we come to choroideremia, I just wrote a grant with a group in Denmark to try to do the kind of work that will be needed for restoring vision in choroideremia. And so that's a 5-year grant that would end in a clinical trial. So we think if we're funded, this is a big grant. This is a $7 million grant that would allow 4 different groups to work together for 5 years to try to achieve this goal, including doing non-human primates. Yeah, I would say, I hate to say that because I know I've been in this field for 25 years and people have been saying 5 years for 25 years. I'm sure many of you in the audience have heard just that. But the take-home message is that these therapies are now in the clinic for a lot of different diseases. Gene therapy and cell therapy is in the clinic for retinitis pigmentosa. And so adapting it to diseases where multiple cell types die is going to take longer. It's going to take a while, but I think I would be shocked if within 5 years there was not a cell therapy for choroideremia. Gene therapy, many of you know more better than I do the experience with gene therapy. There's some unanswered questions there, but I think we're getting to the point where phase 1 trials are going to happen.
We're not going to get into the specifics of the gene therapy trials because a lot of our folks have already heard webinars on that. But since you mentioned that, an anonymous attendee, we have a couple questions coming in,
Okay.
is asking what kind of endpoints might be best for a trial of this nature?
For in choroideremia?
I
Or
would
is
think
it-
so. Yeah, I'm gonna guess that. He didn't specify or she didn't specify that.
So I really do like what Spark has done, where they use these call it a maze, call it a mobility test. I think that's as long as the visual acuity and visual sensation of the patient is within the border zone, shall we say, of a maze test, I think that's really good. Everyday mobility is really important. But beyond that, we use things like microperimetry that just shows what regions of the retina can see. But the ultimate goal is to improve visual acuity. So visual acuity testing, an eye chart, as long as we're in that range, that's what we want to see. We want patients to be able to read and see letters. That's really important.
Nike is asking, is there a plan to do a procedure in the earlier stages as you discussed? You kind of touched on that already. Is there any other?
Yeah, I would say that's more like a phase 3. So first is a phase 1 where you just look at safety, but kind of pay attention, if you know what I'm saying.
Mhm.
And then a phase 2 where visual acuity and visual function is included, but it's still a small scale, typically a dose escalation. You're going from a small dose to a high dose. And then a phase 3 trial when you really do try to prove that your cells are safe and effective. And that's when earlier patients will be brought into a trial.
And sometimes they do combined phase 1, phase 2.
Yeah, typically there's a 1-2B, it's called. So that's usually like 15 patients, something like maybe 20 patients. Again, with a dose escalation, a big gap between these patients to make sure there's no safety issues. So that takes a couple of years to get through a phase 1, 2A. And then you go to 2B and 3 sometimes, which can be combined. You know, the FDA, I've had pretty good interactions with them. They really want to bring these therapies to patients. You know, you're kind of afraid of them at first, but when you work with them, you realize they are on your side. They want to get a safe treatment for patients. So they're guarding safety, but they're not trying to stand in your way. Quite the opposite. They're trying to help you bring this translational work into patients.
Couple more questions. I didn't ask you this when we first talked. Will you be providing your slides to us that we can share, or would you prefer not?
Let me think about that. I think this is all, yeah, I'm happy to do that. That's fine. Yeah, I can do that.
Okay.
Yeah, none of this is confidential.
And then secondly, and another question from Mary, could you track cell fate after intravitreal injection in a pig model, for example, with iron nanoparticles?
So if you notice or not, all the cells in the pig model came from a green pig. So it's easy in a pig. So we have a group in Missouri, Randy Prather's group, Kristen Whitberg that works with us. They made a green pig that has the green fluorescent protein in it. So it's easy to do in a pig. What's hard is the human and whether those iron nanoparticles could be used in it. It's going to happen someday. But I think we need a more robust signal that makes that cost benefit analysis, the safety, efficacy, benefit worthwhile.
The same person that asked anonymously the previous question is asking another one. So since this type of therapy could have potential for individuals with many different types of vision loss, would a phase 1 trial be a basket study? Might it include patients with CHM, or are you considering another specific eye disease?
I would say at this stage, we're looking at diseases where just one cell is lost first. So again, in choroideremia, not only are the RPE cells and the photoreceptors lost, but maybe even Brooks membrane and choriocapillaris are involved. So lots of eye structures are involved. I feel comfortable promoting a tissue engineering approach for choroideremia. That's what I would like to explore for choroideremia. I don't think, so like macular degeneration, just putting in rods and cones into a disease model, it's like moving into a bad neighborhood. You know, you're in trouble. You're going to get robbed. You know, what you want to do is fix the neighborhood and then move in. That's what you want to do to survive. And so you really want to change the local microenvironment. And in choroideremia, that means either repairing through gene therapy, okay? Here's another possibility, is a gene therapy approach that's curative or at least reparative. And then the neighborhood's better. And then the cells might be able to be transplanted, one cell type. So in this case, for choroideremia, I would suggest a gene therapy approach for the RPE. The disease is halted, but vision is still poor. Then a photoreceptor transplant makes sense.
You know, we did hear something, a multi-step, multifaceted approach at our conference, that it wouldn't be just a single type of smoking gun that's going to do it. If I may say, it'd be a multiple.
Yeah.
All right, folks, anything else? I'm going to give you about another minute. If anybody has a question, you can use either the chat or the Q&A, but this has been great. Thank you so much. I think we're almost, I don't see any more typing.
All right. Thank you all so much. I hope this was helpful to people. Yeah, I think this is work that has been supported in the past by the foundation. It's really gratifying to be able to share this with you all, and hoping for good things in the future.
Absolutely. And, you know, we are here always. Everybody's saying thank you so much. We have, you know, a very robust SAB. We have a great community around us. We have fundraising taking place. So please let us know how we can help.
Very good. Thank you so much.
All right. Thanks, Dr. Young. Bye-bye.
Bye-bye.
Thanks, everyone. See you later.